Are you a startup or maybe a university spin-off with a great new therapy, but suddenly you have to follow all kinds of rules regarding release of your product? The regulatory requirements can be quite overwhelming when you're confronted with them for the first time, so you might think about getting rid of the problem by giving your testing to a contract lab. Or maybe you are a larger company but don't have that many samples, so it's not worthwhile for you to establish or keep doing in-house testing. Whatever the reason, if you decide to work with a contract testing lab, you should still understand what's required of you and what your contract lab actually does. And if you are the contract lab, you obviously should know what you are doing as well. With that, welcome everyone to today's webinar where we'll talk about endotoxin and pyrogen testing using contract testing labs. My name is Saskia Ihler. I'm the manager of Lonza's scientific support team in Europe, and I will be your moderator today. Within the next hours, you will get a general overview of what endotoxin and pyrogen testing entails, what is the difference between the two, some basic information about the regulations around testing, and an overview of what challenges you might face when establishing your procedures. And the person who will give you those valuable insights is my dear colleague, Ellen Bergensen. Hi, Ellen.
Hi, Saskia.
Alan is our global subject matter expert on endotoxin and pyrogen testing, and he's been with Lonza for 18 years and for a long time worked in regulatory affairs. He's already worked in the industry regulated by the FDA before, so he has a large wealth of knowledge and nearly 4 decades of experience. And on top of that, he also co-authored a number of well-known regulatory documents on endotoxin and other topics. With all that experience, I am very happy that he'll share his knowledge with us today. However, before we get to the presentation, a little bit of housekeeping information. Our webinar will be followed by a Q&A session. Therefore, please submit your questions at any time during the webinar by using the questions feature. We will address as many questions as possible during the Q&A. And for further information, please also have a look in the Resources tab at the upper right corner of the screen. We are also recording this event, and you will be notified via email once the presentation becomes available for on-demand viewing. So please make yourselves comfortable, grab a coffee or tea or whatever you like, and let's get started. Alan, the stage is yours.
Thank you, Saskia, and welcome everybody. First, we'll start with an introduction. For the labs, we'll talk about quality agreements and working together. We'll talk about the products under test, whether they be drug products, biological products, or medical devices. We'll discuss what a pyrogen is and the various assays. We'll work on setting endotoxin limits, the routine LAL assay, in-process and raw material testing and setting limits there, interferences and how to overcome them, and also the special case of LER, or low endotoxin recovery. So for the labs, a contract testing lab provides testing services to the pharmaceutical, medical device, and other industries. And it's not usually affiliated with the sponsor. However, it could be a subdivision, but it's usually not. They must be registered with the FDA as a contract testing laboratory. They're registered to national and international standards such as ISO 9001, ISO 13485, and if they're a clinical testing lab, they are licensed as CLIA certified. They must follow the applicable regulations for testing, which would be 21 CFR Section 58 for preclinical testing, 21 CFR 211, 610, 820, and also the EMA GLPs in the EU, EMA Annex EP guidance, and also the PIC/S. They must follow applicable chapters in the compendia, and that would be the USP, the EP, the JP, and also the AOAC and others. And for guidelines, The FDA drug and device guidelines are always applicable. The AAMI ST72 guidance document for medical devices, and also the manufacturer's package inserts. If you're working with a contract testing laboratory, you should always have a quality agreement with you and them. It tells you what is expected of them, and it tells them what is expected of you. So the sponsor is ultimately responsible for the testing results and their validity. So you must know how the assay runs, even if you are sending it out to a contract testing laboratory. You have to insist on raw material or raw data being provided, and the assay validation if the assay validation was done for the product qualification. If the assay parameters or performance was altered, ensure the validation covers that change, and sometimes you will have to adjust the different parameters of the assay. So anytime you do that, you have to make sure that that is a valid change. And you should also audit the contract testing lab and qualify them before using to release product to the clinic or to the market. And the sponsor should have a written quality agreement with the with the contract testing laboratory. And the agreement assigns responsibility for different aspects of the testing and reporting process, such as if there was an error or if there was an out of tolerance, they must notify you, but it also tells you when they must notify you. So we've got many different types of products that are under test. We've got drugs and the subset of drugs, biologics, medical devices. There were ancillary products that may go along with those products, such as diluents or other types of products. Oftentimes, you will test your raw materials to know that you've got a low pyro burden going into the final manufacturing product. And also, all of your process water must be tested. So for drugs, the definition for drugs, you can see there under 21 U.S. Code 321 definitions. The term drug means articles recognized by the USP, Official Homeopathic Pharmacopoeia of the United States, or other national formulary, or any supplement to them. So other regions have similar definitions for drugs. Biological products include a wide range of products such as vaccines, blood components, allergenics, somatic cells, and the latest types of products, gene therapies, tissues, and recombinant therapeutic proteins. Biologics can be composed of sugars, proteins, or nucleic acids, or complex combinations of these substances, such as cells and tissues. Biologics are isolated from a variety of natural sources, such as humans, animals, or microorganisms, and may be produced by biotechnology methods and other cutting-edge technologies. So how are biologics different from conventional drugs? In contrast to most drugs that are chemically synthesized with a known chemical structure, most biologics are complex mixtures that are not easily identified or characterized. Biological products, including those manufactured by biotechnology, tend to be heat sensitive and susceptible to microbial contamination, necessitating the use of aseptic principles from the initial manufacturing steps to the final product. Biological products often represent the cutting edge of biomedical research, and in time may offer effective means to treat a variety of medical illnesses and conditions that presently have no other treatment options. So for medical devices in the U.S., they're defined under 21 U.S. Code 321, and a device is an instrument, apparatus, implement, machine, contrivance, implant, in vitro reagent, or other similar related article including any component, part, accessory, which is again recognized in a national formulary such as the United States Pharmacopoeia, intended for the use in the diagnosis of a disease or other conditions, or the cure, mitigation, treatment, or prevention of disease in man or other animals. And they're intended to affect the structure of the body without any chemical action. For example, syringes or medical devices, indwelling catheters, stents, and other regions around the world have similar definitions. So general testing requirements for drugs and devices, and we'll talk with this presentation, endotoxin and pyrogen testing. Parental preparations and implantable devices are routinely tested for pyrogen content before distribution to the public, and that's by law. Test methods used for routine quality control of parenteral drugs are intended to monitor production consistency and to ensure comparability between commercial batches and those batches that were found to be safe and efficacious during the clinical trials. Inherent variability of in vivo assays make them less suitable to ensure potency and safety as compared to appropriately designed in vitro assays. So it's always a better bet to use an in vitro assay where you can. So let's define a pyrogen. Parenteral pharmaceutical products must be free of pyrogenic contaminants. And a pyrogen is linked to an immune defense mechanism and induces fever. Pyrogens stimulate monocytes through toll-like receptors, or TLRs, and activated monocytes release pro-inflammatory cytokines that initiate the febrile response. And as you can see in the graphic in the center, the pyrogen contacts the toll-like receptor, or the TLR, and the monocyte is activated, yielding many different cytokines, among them IL-1 and IL-6, which generate fever with their effect on the hypothalamus and then generate the fever. So what kind of pyrogens do we have? Well, there's the main one, bacterial endotoxin, which is also called lipopolysaccharide, and it reacts with TLR4 on the cell surface. But there is also non-endotoxin pyrogens, or NEPs, and they are the bacterial cell wall, such as peptidoglycan or lipopeptides and flagellin, and you can see the toll-like receptors that are associated with those. There's also mycoplasma, and that's TLR2 and TLR6. Yeasts and molds also have the ability to cause fever, and they, again, react with TLR2 and TLR6. And viruses can stimulate an immune response leading to fever by contact with TLR7 and TLR8. And then there are chemical pyrogens, and they run a wide range of cell surface receptors. So the current compendial tests, and we'll look at some test specificities and limits of detection. To the far left, you'll see a dark blue column for endotoxin and pyrogens, and then across the top, test type, detection mechanism, LOD, and identification. So our first test type is the rabbit pyrogen test, and it can be found in currently, but it will soon be removed, EP 2.6.8, and it's also in the United States Pharmacopoeia under Chapter 151. It's an in vivo measurement using body temperature, and has a limit of detection of approximately 0.5 EUs per ml. The replacement in Europe and proposed replacement in the rest of the world is the monocyte activation test, which measures cytokines released by monocytes from human blood, and that has a lower limit of detection of 0.02. So both of these detect bacterial endotoxins and non-endotoxin pyrogens, such as gram-positive bacteria, yeast, molds, viruses, and etc. But when we get down to the endotoxin-specific assays, we've got limulus amoebocyte lysate, or LAL, and this is compendial all around the world, including EP 2.6.14 as well as USP 85. It's an in vitro measurement measuring initiation of a clotting cascade by the horseshoe crab blood monocytes, or amoebocytes rather. It has a limit of detection even lower than the previous two of 0.005. And the latest assay on the scene is the recombinant factor C assay, which is compendial this year in the European Pharmacopoeia under 2.6.32, and is also in the proposed informational chapter 1085 in the USP. It's a recombinant alternative to LAL, and it's based on factor C, the first component in the LAL cascade. It also has a limit of detection of 0.005, and both of these assays detect gram-negative bacterial endotoxin. And this graph shows the comparability between all of the different assays. If you go to the far left, you can see from the 1970s until current. The initial assay was the gel clot assay, and as you can see in that column, every LAL assay and recombinant assay begins with factor C. Now, in the gel clot assay, factor C reacts with factor B, producing a proclotting enzyme, which it affects the proclotting enzyme, making a clotting enzyme, which acts on coagulagen to make coagulin, giving you a gel clot. So if you wanted to make a kinetic assay out of that, you would simply measure the time it takes for a turbidity to cross the OD threshold, and so the more endotoxin, the shorter the time. And again, you can see the assay steps are exactly the same, except this time you're measuring the time it takes for the turbidity to cross the threshold. Going into chromogenic assays, it's the same chemical cascade, except instead of cleaving coagulagen into coagulin, you'll cleave a chromogenic substrate and you'll get a yellow color, which is proportional to the amount of endotoxin in the assay. But to make a kinetic assay out of that, again, it's the same exact enzymatic reaction where you'll cleave the chromogenic substrate, but now you're measuring the time it takes for that yellow color to cross the OD threshold. So it's the same as the turbidimetric assay. But now coming to recombinant factor C, all those other steps you see in the middle column are all amplification steps. But with a factor C assay, you don't need any signal amplification steps because you're generating light, and light is much easier to measure. So the factor C, when it contacts endotoxin, will cleave the fluorogenic substrate, and when you excite it at 380 nanometers, you will get fluorescence at 400 nanometers, and so you're measuring light output, which is proportional to the amount of endotoxin in the product. So why are two tests for pyrogenicity recommended? Non-endotoxin pyrogens cannot be detected with the BET. And remember, you have to have account for any pyrogen that may come into your product. And so during your preclinical studies, this is where you will use another method. So the BET is less complex, cheaper, and faster for routine testing, and more importantly, process control. So you'll use this for in-process as well as final product testing. However, the LAL test contains certain limitations. It doesn't detect non-endotoxin pyrogens such as lipoproteins, peptidoglycan, lipoteichoic acids from gram-positive bacteria, and therefore it's an imperfect reflection of the human response. So the evidence must be provided that especially adjuvanted vaccine formulations do not interfere with the LAL test. Consequently, it's recommended to supplement the LAL test's ability to detect low levels of endotoxin with a specific test for non-endotoxin pyrogens. And the MAT assay is complementary to the BET and can be used at specific points in the manufacturing process. And as you can see in the graphic to the right, we have for method endotoxin and non-endotoxin pyrogens, and the targets, and the number of diamonds indicates the level of suggestion for using that assay. So for endotoxin, the BET is highly recommended. But for other bacteria, the MAT is highly recommended. Also viruses and yeast and molds. Now we're going to look at testing parenteral products for pyrogenicity. And we'll look at the regulatory chapters and the information that the tests provide. Drug products are regulated in the U.S. under the authority of the Food, Drug, and Cosmetic Act by the U.S. Food and Drug Administration. The regulations may be found in Title 21 of the U.S. Code of Federal Regulations under 21 CFR 211. It states that for each batch of drug product purporting to be sterile and/or pyrogen-free, there shall be appropriate laboratory testing to determine conformance to such requirements. The test procedures shall be written and shall be followed. And so that's for the FDA. However, if you are making animal vaccines, they're regulated in the U.S. by the U.S. Department of Agriculture, but you still have the same requirements of non-pyrogenicity. And there are also similar requirements for non-pyrogenic devices. And other regions have similar regulations, such as in Europe, the EMA, in Great Britain, the MHRA, in Japan, the PMDA, in Brazil, ANVISA, and in Australia, TGA, and there are many, many other individual country organizations. So let's look at the U.S. regulations. In the U.S., parenteral drugs are regulated by FDA's Center for Biologics Evaluation and Research, the Center for Drug Evaluation and Research, and the Center for Devices and Radiological Health, or CDRH. All injected products are required to be tested for pyrogenicity unless the product is specifically exempted by regulation. And the official test in the United States still for biologics is the rabbit test, the rabbit pyrogen test. And you can see below that the specifications for the rabbit pyrogen test. But with a lot of products, you cannot test in rabbits because you'll have an adverse event in the rabbit. In such case, you do get an exemption. However, that's exemption from the rabbit pyrogen test. That is not exemption from the LAL test or the MAT test. So under 61013, General Biological Products Standards and Purity, it gives you the requirement, the actual requirement, and as you can see, the legal requirement is intravenous injection into rabbits, and it also gives you all of the exemptions. So if you're making a vaccine that is going to be detrimental to the health of the rabbit, you do have an exemption from the rabbit. However, you still do have to do the LAL or MAT assays. In the EU, the EU Directive 2010/63 regulates the use of experimental animals for science and quality control purposes, and you have to look for a replacement of an in vivo test with an in vitro test. Chapter 5.214, guidance for substitution of in vivo methods, outlines the importance to consider in establishing in vitro methods for quality control during vaccine development and for human and veterinary use. And again, it states the inherent variability of in vivo assays can make them less suitable than appropriately designed in vitro assays for monitoring consistency of production, for assessing potential impact of manufacturing changes. And in Europe, again, we have the EMA, and this Committee for Medical Products for Human Use, the CHMP, the Committee for Medicinal Products for Veterinary Use, or the CVMP. And there are also alternatives to the RPT, such as the MAT, and you must consider them, and that is found in EP 5.1.10, EP 2.6.14, EP 2.6.30 for the monocyte activation test, and now 2.6.32, which was made an official compendial assay in January 1st of this year, and that's for recombinant factor C. If we're looking to Asia, there are minimum requirements for testing biologics, and they're established by the National Institutes of Infectious Disease in Japan, for example. And they also require the use of the rabbit pyrogen test. And their rabbit pyrogen test is a little more complex, but as you can see down below what the requirements are and the specifications. And you follow Chapter 4.01 bacterial endotoxins for drugs and medical devices if you do not use the rabbit pyrogen test. In China, biologic testing requirements are found in the Chinese Pharmacopoeia under general requirements for preparation/injections. Parenteral drugs in general should be tested for endotoxin and pyrogens unless with special reasons for exemption. And the endotoxin level of the raw materials and the excipients that are used in the manufacturing process should also be tested and controlled. And that is a departure from the other compendia. So endotoxin content and non-endotoxin pyrogenicity. Parenteral products and implantable medical devices are routinely tested for endotoxin using LAL before distribution to the public, as we've already discussed. Viral vaccines need not be tested in rabbits for pyrogenicity if the vaccine may be incompatible with the rabbit, causing the rabbit to get sick or die. Vaccines that are meant to stimulate the immune system and cause a fever in the animal unrelated to a pyrogen. In general, the rabbit pyrogen shall be replaced in the EU following EU directives. And the MAT is the in vitro replacement for the rabbit pyrogen test in Europe. And vaccine components, it must be noted, may trigger the cells used in the MAT to release cytokines. So you would use the method C batch comparative test to test vaccine batches with reference lots that are clinically tested. So in comparison of the rabbit pyrogen test versus the monocyte activation test versus LAL, we can look at the regulatory chapters. Both the MAT and the rabbit pyrogen test provide information about the biological impact of the sample. They are essentially a form of dose response assay. The bacterial endotoxin test, or BET, determines the quantifiable endotoxin concentration in a sample and is not a direct activity, although they are closely related. Due to its speed and precision, the BET is additionally valuable as an in-process control. And the MAT mimics the overall immune response, and that because it uses human cells. And again, we can see the test chapters. We've got the rabbit pyrogen test. It's non-specific. It's a temperature increase in rabbits. The time to result is greater than 1 day. It's actually a 2-day assay. Your preclinical, you'll use it there for end product if applicable, and some regulators are making you use the rabbit pyrogen test if you have LER, and we'll get to that towards the end of this presentation. You won't use it as an in-process test because it really takes too long, and what you're looking for is the effect. So with the BET recombinant factor C, it's specific for endotoxin. It's initial activation of factor C. It takes less than 2 hours to perform. You will use it preclinically for end product testing, in process testing, and it's the presence of endotoxin. And for the monocyte activation test under 2.6.30, again, it's nonspecific. It's looking for the release of pro-inflammatory cytokines. It takes greater than 1 day, and it's, again, actually a 2-day assay. You'll use it for preclinicals, and you could use it for end product testing, and you'd also use it after you've done the comparability and validation, and where you switch over to LAL or RFC, if you have a process change that you think may generate a non-endotoxin pyrogen, such as any cytokine. And you're looking here at an effect. Testing of materials for parental drug or device manufacturer. What you have to remember is that the endotoxin contributions to the final product is really a sum of the parts of all your raw materials and process. So in order to minimize the endotoxin concentration from various components and processes that are involved in manufacturing of a biologic product, testing of the following is needed. You should test all of your raw materials, all of your formulation water. You should be testing your in-process intermediates to look for contamination, and also if it's a endotoxin reduction step, to make sure that it in fact did reduce the endotoxin, and also your final vialed product or drug product. And once again, the overall endotoxin burden will be the composite of the individual contributions. So this is a schematic of a general manufacturing process, whether it be a biologic, a cell therapy product, or monoclonal antibodies, or a vaccine. First, you'll start with your media prep for your seed stock, and you'll develop a working cell bank, and you'll do a lot of subcultures, and then you'll inoculate the next step up, which could be shaker flasks or a wave bag, and then you'll move up to larger fermenters as you scale up, and then you will centrifuge, depth filter, and then collect. So the two strongly suggested places to do your endotoxin testing is during your media prep, and also your bioburden and endotoxin at the end of the process. However, it's good practice to be testing between each of those process steps to make sure that you don't have an introduced contamination. So now that we've got our bulk, now we have to do our purification process. And again, this is a representative schematic, could be any type of biologic manufacturing process. But you have your harvest collection that you'll put over a chromatography skid through a column, and then you'll collect your eluate, and you should be doing a bioburden and endotoxin assays there. And then your next chromatography skid, you'll do the same, and when you're done, you'll again do bioburden and endotoxin. And then the next chromatography step, you'll again, after you go through the step and go to your holding tank, you'll do bioburden and endotoxin there. And then you just keep doing that for every purification step that you have until you get to your bulk drug substance. Now, this is where you will add, in the case of biologics such as monoclonal antibodies, you'll add citrate in a polysorbate, and this is where the LER problem comes in. So now, moving on to cell therapy products. Cell therapy products are generally manufactured in smaller batches, and they most times are one batch, one patient. So with that, unless you're making stem cells, and in this example, we'll look at some stem cells. You'll collect cord blood, you'll select for the CD34 cells, you'll expand your CD34 plus cells, you'll transform them into HIPSEs, you'll create a master cell bank, and you'll create a working cell bank. And then from there, you'll expand the working cell bank, and you'll transform into the desired cell type. You'll expand those cells, you'll wash them, and you'll create a dosage form for a patient, and you'll administer to that patient. So where should you be testing all along that process? As you can see, just about every in-process intermediate you'll be testing, because this process is generally open. You'll be doing them in a hood or a biosafety cabinet, yet you still have open manipulation. So you really want to do bioburden and endotoxin detection at each of those. Number one, for patient safety. Number two, if you have a contamination, you can stop the process right then and there and not waste time and effort, and you can start over. So the steps to routine testing, you must, if you're the contract manufacturer, if you're the manufacturer or the contract test lab, you must get the product information and set specifications. And you must establish an endotoxin release limit. You must calculate the maximum valid dilution. You perform your inhibition and enhancement testing to establish your minimal inhibitory concentration. And then from there, you'll choose a product dilution, and you'll do your product validation. And then from there, you'll go into routine testing. So for parenteral product testing, we'll talk about setting specifications, and we'll use, for an example, a small volume parenteral of less than 25 milliliters delivered in a syringe with direct injection. And we'll also look at a cell therapy large volume parenteral, which is greater than 25 milliliters, and that's something that would be administered via infusion through a blood bag. So for our small product testing, you have to set your endotoxin release limit. And that is in all of the compendia established by K/M, where K is the threshold pyrogenic dose for humans, which is 5 endotoxin units per kilogram per hour. And don't forget that per hour in red. And if you're looking at intrathecals, it's 0.2 EU per kilogram per hour. And M is the maximum total dose delivered in a single hour. So for that, you take the calculations and you multiply 5 EU per kilogram by the dose, or by the mass of the patient, and ordinarily it's approximately 70 kilograms. And we were saying that we were looking at a 2.5 milliliter dose. So that would give you 140 endotoxin units per milliliter as your endotoxin release limit. For your maximum valid dilution, it's the endotoxin release limit multiplied by the concentration of the solution, which is usually 1, divided by lambda, or the sensitivity of your assay. Now, in the case of chromogenic and turbidimetric assays, it could be anywhere from 0.01 to 0.001, or for our chromogenic assay, it is 0.005. So the maximum valid dilution in this instance is 140 divided by 0.005 for our chromogenic assay to give you 1 to 28,000. So you can have a very good dilution scheme to overcome any problems that you may find. So with testing medical devices, the compendial endotoxin release limit for medical devices is 20 EUs per device. Medical devices are tested as per USP 161 medical devices, bacterial endotoxin, and pyrogen tests. Also, ANSI or AMI, ST72, bacterial endotoxin test methods, routine monitoring, and alternatives to batch testing is another guidance document. And for these assays, the devices are typically soaked in LAL reagent water for 60 minutes at 37 degrees, and the endotoxin is measured in the extraction fluid. Extracting LAL reagent water volume may be altered to accommodate various device sizes. So it makes very little sense to use a full 40 mLs for a tiny device, nor does it make sense to use 40 mLs for a huge medical device. And the endotoxin release limit is K times N divided by V, where K is the number of allowable endotoxin per device, which is 20. N is the number of devices, and V is the extractant LAL reagent water volume. If the entire device is implanted, all surfaces must be tested. That means that the entire device must be immersed in LAL reagent water for 1 hour at 37 C. However, if you have a syringe or a device with a fluid path or lumen, the entire fluid path must be extracted, not the entire device. So you will fill the lumen of that device with LAL reagent water and then incubate it and then test the fluid. And as you can see, the examples here of something with a lumen are the syringes on the top. And if it's implanted, these are stents, cardiac or biliary stents. So because these are implanted completely, you would immerse the entire product. So now we can look at interferences. What will interfere with the assays? First, you have to do your validation of spike recover. So you have to know your endotoxin release limit, your maximum valid dilution, and you'll perform a dilution series all the way out to your maximum valid dilution. And you'll spike each dilution with a CSE, which is a controlled standard endotoxin, or the reference standard endotoxin to the middle of the standard curve, which is typically 0.5 to 0.05 endotoxin units per ml. And a valid recovery is recovering your PPC, your positive product control, between 50 and 200% of that spike. And this shows what you should aim for in your product spikes. You're looking for interferences. Now, they could be inhibition, where you're not recovering your spike, or enhancement, where there are times when the apparent endotoxin content is elevated due to enhancement and from things like beta-glucans. It's determined by judging the recovery of a known endotoxin spike, a positive product control, or PPC. And you again dilute all the way out to your MVD, spike your PPC in the middle of the standard curve for those dilutions. But there is a special procedure for the gel clot that we won't get into. And the specification of PPC recovery is 50 to 200%. Now, although you're allowed 50 to 200%, it's really not recommended to accept any values at those extremes because of the variability of the assay. So the closer you get to the extremes, the higher the probability of you having an assay that passes a product that potentially could have failed. So you're really looking at the green areas between 70 and 160%. That's ideal. With borderline, you can have between 160 and 180%, or between 60 and 70%. And only in rare cases where you really can't get an acceptable spike recovery should you accept 180 to 200 or 50 to 60%. So the different levels of interference that we have here. So if we're looking at the endotoxin, it could be affected by solubility. The endotoxin could have formed micelles in the solution. Binding, endotoxin does bind to things. Aggregation, again, it's a micelle issue. And there's also masking, which comes in with LER. for our enzymatic cascade, that can be affected by the pH of the solution, which is why you must always have a pH between 6 and 8, and you should try to optimize towards neutrality or 7. Ionic strength. If you have a high salt content or a low salt content product, you may have issues with the enzymatic cascade performing. Mostly high salt concentrations, though. You can have depletion of divalent cations with the use of chelators and other agents that will remove magnesium or calcium from your solution, in which case you have to add them back. There could be enzymatic activity in the sample, such as proteases, which will also cleave your substrate. And you can also have enzyme inhibition from various things in your product. For detection, if you have a very high background of endotoxin, it will be very difficult to recover a low level of spike in your endotoxin. So you're going to have to dilute out closer to your MBD with such a high background. You can have precipitation, and vibration also is not recommended for doing an LAL assay because it interferes with the gel clot formation or turbidity formation. And you also have calculation errors due to lab issues that you have to be aware of. So to overcome interferences, we're looking at dilution, and you can do that with most samples up to your MBD. You can adjust the pH. You can use unbuffered samples with a pH outside of the range if that's the case, and you must dilute to pH 6 to 8, and that is in the sample lysate mixture, not the sample alone, because the lysate itself does have buffering capability. You can inactivate with heat, and that would be for samples containing active proteases or proteins causing endotoxin masking. And there are a variety of times and temperatures, but what we recommend is 75 C for 30 minutes. Surfactant dispersing agents such as pyrosperse can be used, and that would be for oily samples or samples that bind that endotoxin, and also partially with micelles. You can use glucan blockers to combat LAL reactive material such as beta glucans. And you can also do divalent cation addition, and we recommend magnesium chloride to counteract chelators such as EDTA and citrates. So if you were doing your spike recovery and you see this result, the first result that you come to that is acceptable, quote unquote acceptable, is 1 to 50. However, we wouldn't recommend that because it's right at the edge. 75 is good, but 500 is probably better because your maximum valid dilution is 2800, and this gives you closer to 100%. And as you can see, this is what we would recommend. So, validation of your spike recovery. Anytime you do anything other than dilution with water, you have to validate that process. So you'll spike, you'll treat, and you'll test. And so one of the treatments could be adding pyrosperse, spike, and then for a serum sample, incubate for 70 degrees C for 10 minutes, and then you'll test. One thing you do have to be aware of that some treatments cause denaturation of proteins. And when denaturation of proteins happens, the endotoxin actually gets locked up in that protein. And so you might not be able to recover your spike. So now we'll get to the issue of low endotoxin recovery. LER is a controversial topic that's been circulating since 2013 when it was brought to FDA's attention by Dr. Joseph Chen. It's theoretically described as a masking effect manifested in biophysical formation of a complex that blocks the ability of factor C to bind to endotoxin. So if factor C cannot see the endotoxin, it can't react with it. What also must be understood, though, if factor C can't see it, neither can TLR4 on the cells. And LER subjected samples also show poor MAT variability for the same reason. The cell surface receptors cannot see it. So where does it occur? It's primarily in biologics manufacturing, where you'll use polysorbate combined with a chelator, or such as citrate or phosphate. Polysorbate is estimated to be in more than 70% of protein formulations nowadays. There's also some evidence that histidine formulations cause the issue, and LER is considered to be distinct from interferences commonly seen with the LAL assay that can usually be easily overcome, because LER is not easily overcome. So with LER, remember, it's a two-part issue. So if you just have sodium citrate in a sample and you do your spike, over time the spike remains fairly stable, and that's seen in orange here. If you just have polysorbate 20 and you spike, over time you'll see it should be fairly stable or slightly rise. That's because of the dispersion characteristics of the surfactant, but it should stay fairly stable. However, if you have both polysorbate and sodium citrate in your solution, you'll spike, and then over time the apparent endotoxin levels appear to fall. And I should say that is apparent endotoxin, because the endotoxin is still there. It's just bound up inside of a micelle. Now, LER is also highly temperature dependent. And as you can see here, at 2 to 8 degrees C, if you spike it and you follow the top line, it takes outwards of 16 hours to actually have a 50% reduction in the amount of endotoxin in the product. However, if you go to ambient temperature, it happens fairly rapidly. And if you go to 37 degrees C, it's even faster, and it occurs in minutes. So basic facts: LER is dilution dependent. It cannot be overcome by sample dilution because it forms an extremely stable structure that you can't reverse. However, what we describe as LER is not often seen in the endotoxin that may contaminate our products. In this graph, you'll see a sample of a monoclonal antibody in 20 millimolar citrate with polysorbate, and a natural occurring endotoxin was easily recoverable. However, the RSE or the CSE was not, and that's because of the structure of RSE and CSE. They are highly modified structures due to the extraction of endotoxin and then purification process with hot phenol, and hot phenol strips away the polysaccharide tail, yielding a highly hydrophobic molecule, and it disappears due to LER. So what's the current regulatory stance? That the FDA included a stability screen in its 2012 Q&A guidance document, and it recommended that the industry perform hold time studies to verify the amount of time a sample can be reasonably stored before final release testing. And the products showing LER that cannot be overcome must be tested by the rapid pyrogen test. Okay, the rapid pyrogen test requirement applies to all of the BLA products regulated by CBER and CDER. For non-biologics, aka small molecules, equivalence is assumed between the rapid pyrogen test and the BET. So in lieu of the rapid pyrogen test, endotoxins can be used if the method is shown to be equivalent under 21 CFR 610.9, where you have to show evidence demonstrating the modification will provide assurances to the safety, purity, accuracy, and effectiveness of the biological product that is equal to or greater than the rabbit pyrogen test. So FDA CDER is currently requesting follow-up studies post-approval for drugs exhibiting LER. So why mostly with biologics? Because the BLA is a formal request for permission to introduce or deliver for introduction into commerce for biologics, and because it's mostly seen in biologics, the FDA is requesting it for biologics. And if the information meets the FDA requirements, the application is approved and a license is issued, allowing you to sell your product on the market. And once again, biologics are most likely to contain polysorbates and a chelator for stability. So the mechanism, this mechanism was developed by Johannes Reich of Hyglos. Now, and so you have lipopolysaccharides, and they are bound together with magnesium divalent cations, and this makes the cell membrane, and so it's a highly stable matrix. So if you have a surfactant and you introduce it, it doesn't do anything to this matrix. However, if you add a chelator, such as sodium citrate or EDTA, the chelator picks out the magnesium divalent cations and replaces them with sodiums. So as you can see here, the sodium ions are attached to the phosphates of the lipopolysaccharides, and it allows the surfactants to get into the membrane and gradually dissolve it. And as you can see, it progresses even more, and the endotoxin is surrounded by the sodiums, but in between them, you're getting more and more surfactants, until finally you have a monomer of endotoxin surrounded by a micelle of surfactant, in this case polysorbate 20. So you'll get inadequate detection because the lipids, the lipid moiety cannot be seen because the acylations are on the inside of the micelle, and it's the acylations that interact with factor C. So this essentially detoxifies the endotoxin molecule. So how to recognize it? You have to do a screening. So if you're sending your products to a contract testing lab, you must be aware that they will have to do an LER whole time screening to try to overcome it. And it's a simple test using RSE and CSE. And you have to ask yourself, does my product contain polysorbate? And that's the trigger. And also, does it also contain a chelator? And you spike the undiluted drug product or in-process solution with endotoxin. The spike should be at the midpoint of the standard curve after dilution. And the CSE is recommended for LER screening as a standard. And you should carry out whole time studies to determine whether initial spikes are recoverable. And so before you submit your product to your contract test laboratory, or if you are the contract test laboratory, you must be aware of this. So again, you directly spike into the drug product. You hold the product at a process significant temperature over time, and you test at specific time points out to the amount of time that you think it would take from your final formulation until an actual laboratory endotoxin test happens. And you determine the percent recovery of the spike relative to the amount of endotoxin originally added to the sample. So, with that said, if we can satisfy your testing needs, whether you are the contract test lab or whether you are the sponsor. We have our endotoxin and pyrogen testing, including our pyrogen gel cloud assays, our pyrogen 5000 turbidimetric LAL assays, our kinetic QCL chromogenic LAL assays, our pyrogen recombinant factor C assays, as well as our new Pyrocell MAT system, along with all your reference standards, consumables, instrumentation, and robotic solutions, our informatics, as well as our other services. And we have our monocyte activation test, which is new. It combines the Pyrocell MAT test kit and the Pellikine human IL-6 ELISA kit for 96 well plates, and it uses pooled cryopreserved cells that are in optimized MAT culture medium, and it comes from quantified or qualified blood donors from an actual blood bank. And the Pellikine assay was the pioneer in the IL-6 assay. It includes all reagents that you'll need, and it's validated with the Pyrocell MAT kit. We also offer the Pyrotech Pro system, and so for contract test labs who are doing high throughput, we highly recommend this, and also for our larger customers. It's a robotic solution for endotoxin assays using the TCAN Freedom Evo platform. It's a plug-in for our WinKQCL 6.0 and above software, which is renowned for its security of its database, its testing engine, and its reader controls. It supports Lonza Kinetic KQCL, Pyrogen 5000, and Pyrogen assays, which we've got literature on. It standardizes the operating procedure to automate all of the manual processes for less variability, and it addresses the needs for data integrity because it's got a customization transfer of data. It eliminates paper forms and the inherent transcription errors, and it automates the data transcription. And in some, injectable drugs and implantable medical devices are critical components in maintaining worldwide human health. And as you can see, the virus to the right is what we are really concerned with right now. Different compendial tests are available. However, inherent pyrogenicity or matrix complexity may not support current in vivo testing methods. Therefore, the BET process for testing for bacterial endotoxin should be complemented by an MAT assay for testing other pyrogens. While similar, regional requirements are not harmonized, leading to complexity of market authorization. So you must know where your drug is going to be going and what type of testing variability there is. It's critical that the sponsor and their contract testing lab maintain constant communication, and the vehicle for that is your quality agreement. It tells you who, what, when, and where of all communication and all responsibilities. Sponsors must have a quality agreement with their CTL that specifies the responsibilities and your actions. Again, the who, what, when, and where. The sponsor must also qualify and audit their contract testing laboratories. Don't just send a sample to a lab and get an assay back. The sponsor must be the ultimate owner of the data if it is ever requested in a regulatory audit. So part of your agreement should be to get not only a cover report but also the raw printouts of your assay, and you should also have product-specific qualification. And with that, we will enter our Q&A session. Saskia?
Yeah, thanks a lot for that, Alan. That's really a lot of ground that we covered here, and we did receive a number of questions throughout the presentation. Unfortunately, we don't really have the time, but there were some that were asked in different variations, so maybe we'll just answer those. And if your question isn't answered live, don't worry, we'll get back to everyone after the webinar within the next few days. So I have tried in the background to kind of group the questions a little bit. So let's see, maybe we get to 2 or 3 at least. So there was a question that was asked for MAT, whether the testing for pyrogenicity is needed for every batch.
When you were in your preclinical studies, yes, you would do the MAT in replacement of the rabbit pyrogen test because you were doing all of your ADME-tox studies at that time. So you have to know all of the different pyrogens that may be in the product before it goes into humans. So you'll do an MAT as well as an MAT in Europe or a rabbit pyrogen test in the United States, and then you'll do it in conjunction with the bacterial endotoxin test, finding no pyrogens, non-endotoxin pyrogens in the preclinical batches. Then you'll move to the BET assay. And as I said before, if you suspect a change or if you come back with a clinical response of pyrogenicity, then you could go back to the MAT as an investigative tool. But for actual routine product release, no, you'd use the BET.
Okay, thanks for that. And we have someone asking how samples should be shipped to contract testing labs.
And that's something that is very critical. The shipping conditions should match your labeled storage conditions unless you have an exemption from the FDA. So that means if your product is labeled for 2 to 8 storage, you should be sending it in a validated shipping container using wet ice packs, but not enough wet ice in order that the product freezes. However, you should have some freezing point depression, so you should be okay, and you may get below 2 degrees on that. If your product is meant to be shipped frozen, you must maintain frozen, and with that, it's usually dry ice. And for extremely sensitive products, you will ship under vapor phase liquid nitrogen, but those are few and far between. But yes, it is critical that you maintain the proper storage conditions and work that out with, if you're a sponsor, work that out with your contract testing lab to make sure that they can handle the storage conditions for your product.
Thanks. I think that is very important information. And maybe one last question. I'm looking at the time again. There is someone who asks whether or not you can send a protocol for overcoming LER. Maybe that question also goes a little bit to me. I don't know. Do you want to take this one?
Sure. The best option is to get a copy of the Parenteral Drug Association or PDA Technical Report 82 on LER. I was actually one of the co-authors of that document, and it goes into all of the different things you can try. There's really no one set protocol for overcoming LER, and one of the issues is you're looking at preventing it before it happens. So you may have to manipulate your sample collection process to introduce something that counteracts the effects. But once LER happens, it's very difficult to overcome. And TR82 from PDA gives you 13 different case studies on different ways to overcome it. And I hope that answers your question. Saskia, do you have anything to add to that?
Yeah. No, I just wanted to say that there were different people asking similar questions for very specific products. But as you said, Ellen, it's really product specific what you have to do. So I would also like to refer everyone to TR82. And I think maybe one last question from someone who says they have done in-house testing, until now, but they want to give their testing to an external lab. What's the best way for the transfer process?
Okay, well, if you've been doing your own internal testing, you already know what your maximum valid dilution is, your minimal inhibitory concentration, and your standard testing dilution. So when you send it to a contract testing lab, they should be able to repeat those parameters. Now, hopefully, you've chosen a contract testing lab that does a lot of endotoxin detection. So if they're qualified, if their analysts are qualified, meaning usually it's a water test and being able to recover successfully a standard curve and a spiked water sample. Then after that, it would be just testing your product. And if you're actually doing a product validation, you're looking at establishing the MIC in their hands and the MBD in their hands, and then looking at testing at your established test dilution. Because if they don't test at your established test conditions, you may see either a raise or a jump or either a jump or a drop in the levels, and all that is due to is because of the dilution. So transfer all of your testing parameters to them and allow them to run the tests as you do.
Okay, thanks a lot, Alan. I'm afraid that's all the time that we have for today. So as I said, if we didn't answer your question, we'll get back to you by email within the next few days. And just as a reminder, you will also receive an email notification once today's presentation becomes available for on-demand viewing. So thanks a lot to everyone for taking the time to join us for this webinar. Have a wonderful day and see you next time. Bye-bye.
Thank you, everybody.